talked about carbohydrates. We spent a whole chapter talking about simple sugars, complex sugars, the polysaccharides, and then we spent sort of like half of 2 chapters talking about lipids. So we talked about phospholipids and about cholesterol, so about like what lipids are used for in the cell. Proteins are the third group. So you have carbohydrates, fats, and proteins. So proteins, this chapter is really talking about like what are proteins made of, what are their structural components, and then goes back again to enzymes and kind of reviews enzymes again. We've already sort of introduced them, but it does spend some time talking about that. So proteins, while polysaccharides, the building blocks are glucose. So like when you're talking about starches, you're talking about fiber, you're talking about large chains of glucose. When talking about proteins, the building blocks are called amino acids. So these are the building blocks of proteins. And their name comes from the amino and the acid. So remember, anything that's amines or amides, they always have what? So remember that in the functional groups? Amines and amines, they have what atom in them?
Nitrogen.
That nitrogen. And so that's the amine side. So every amino acid has a nitrogen. Every amino acid also has a carboxylate or carboxylic acid group. And so those 2 parts, the amino and the acid, is where that name actually came from. So you see these 2 groups on every single building block of proteins, every single amino acid. There are 20 amino acids that are found in most proteins. They differ based on their chemical formula, but of the 20, 8 of them are called essential. So 8 amino acids you have to get in your diet. You can't make them. So that's what an essential amino acid is. For infants, they actually have to have 10, but once you reach adulthood, you actually become functional in making 2 of the other ones. So it's really 8 for an adult. So an essential amino acid is an amino acid that has to be gained in the diet. Your body can't make it. The other 12, though, you can make. You can actually convert one amino acid to another type of amino acid, so you can sort of swap them around and build the amino acids for the other 12. But if you don't get these 8 essential, you'll end up deficient in protein, which means that your body is not going to be able to make protein because it will not have all 20 of the building blocks. So when they talk about a complete protein is one that has all 8 essential. And for the average person with the average diet, what is the most common source of protein in your diet? Meat, okay? And all meat, whether you're talking about beef or pork or chicken or seafood, all meat is a complete protein source. So any and all. So there comes the question of the vegetarians. Well, how do vegetarians survive because they don't eat meat? It's not just beans. Beans are actually missing 3 essential amino acids. And you eat them in combination with grains. Grains are missing 2 of the other essential amino acids. So by eating a combination of them, you can actually get all of your essential amino acids. But you have to have both. So you have to have beans or legumes in your diet. And you also have to have grains in your diet because beans are missing a couple, grains are missing a couple other, but by getting them in combination, you can still have adequate amounts of amino acids to be able to build the proteins that you need. So this is the thing. So there are some countries that meat is really it's really not part of their normal diet just because of refrigeration issues, because of the cost that's associated with it. And so that's why a lot of people will eat things like black beans and rice is sort of a staple South American diet. The rice and beans, peanut butter and jelly is actually a complete protein source. because the peanut butter is a legume, or peanuts are. They're not actually nuts because, they grow in the ground. So they actually are considered a bean, just like black beans and kidney beans. They're considered a bean source. So but plus the bread. So the bread would actually give you the grain source. So breads and cereals along with those grains will end up being able to give you that complete protein source. Other interesting thing about how much protein do you need in a day if you were thinking about a piece of meat? Once a day. Once a day. If you have a piece of meat that's about the size of your palm, that is all of your essential amino acids that would contain everything that you would need to be able to build protein. Now, if you were like a marathon runner, or if you were like working out at the gym, then you might need to up your protein. But in fact, the average American eats like 5 times more protein than they need to. Because haven't you seen steaks that are hanging off of the sides of a plate? And the plate is a platter. It's not actually a plate. They're humongous. But in fact, a piece of meat that's basically like a quarter pounder, that's pretty much all you need. Okay, so it doesn't require large amounts. As long as you're getting the essential 8 amino acids in the diet, then you're fine. Structurally, amino acids all have that nitrogen. Amino acids all have that carboxylate or carboxylic acid side. What makes them different, they all have a hydrogen on their center carbon. And it's always interesting to me, they're NCC, like National Community College. So there's a nitrogen and 2 carbons. So all amino acids will have this nitrogen, carbon, carbon. The end carbon will have 2 oxygens. The middle carbon will have a hydrogen. And so that's always the same. So sometimes they call it like the backbone, because it's exactly the same for all amino acids. What makes them different, what makes one amino acid different from another is this. And if you look on the periodic table, there is no element that says R. There is no such thing as element R. So the word R stands for the group that is unique to that amino acid. So here's looking at the R groups. Let's look at some of the R groups. So this first group, the peachy colored area, are all in common. That is the nitrogen, carbon, carbon group. That they sometimes call the backbone. So the nitrogen, carbon, carbon. This group, this is a positive, this is a negative. That's the same. And then that hydrogen. So that's all exactly the same, every single amino acid that you look at on this page. So there's 9 of them on this page. Every single one has got that peach colored area that's got nitrogen, carbon, carbon, and a hydrogen hanging off of the middle carbon. It's the purple part that's different. So this is what they call the R group. And it varies. So as you look across, you see that each one has a different group hanging off of that middle carbon. All of these are nonpolar. So all of these amino acids have a nonpolar R group. So if we zoom in a little, just so you can look at them a little closer. The first one, glycine, it just has a hydrogen. The second one, alanine, it has a methyl group, a CH3. The third one, valine's got 3 carbons. But notice nothing but carbons and hydrogen. When you have nothing but carbons and hydrogen, you're going to have a nonpolar molecule going down. See, isoleucine's got 4 carbons. Leucine, 4 carbons, just a little different arrangement. Phenylalanine, we actually talked about phenylalanine, and some people don't have the enzyme to break this down. It causes PKU. Phenylalanine, it has an aromatics. Remember, that's that alternating single and double bonds in the molecule, but nothing but carbons and hydrogen. So if you have nothing but carbons and hydrogen, then those are going to be the nonpolar R groups. Now in this one, proline's a little bit different in that proline has that the R group actually reconnects onto the nitrogen that's on that far side, the amine side. That's the only one that does that, but it does form a little bit different. Then there's methionine that's on there as well. Methionine down at that bottom one, and it's different because it's got a sulfur. But remember, there's no nitrogen, oxygen, fluorines, or chlorines on that R group, so these are all considered nonpolar. So these are nonpolar. Go down. Yeah, nonpolar. The R groups is really what you're talking about. The R groups are hydrophobic. Right? Remember, hydrophobic is like water-fearing. All of these R groups are not going to want to interact with water. And that becomes important when you start talking about the shape and the folding. These amino acids on the next page, there's 6 of them, and they have nitrogen oxygens to them. They are polar. Okay, so they're going to want to interact with water. So these polar R groups, hydrophilic, water loving, they want to interact with water, and that's going to affect the shape of the molecule if you have these in there. So in looking at them, so there's tyrosine. So see, it's a ring structure, but see on the end, it's got an alcohol group. Asparagine, it's got a nitrogen and an oxygen. So having nitrogen, oxygens, fluorines, or chlorines are going to increase that polarity. These have oxygen. The one on the top, the sulfur, cysteine, it's polar because it's such a little R group. Remember, little molecules can have a little more polarity. It's not really all that polar, but it's a little bit. And then glutamine has another oxygen and a nitrogen group. It's an amide. Threonine has the alcohol. Serine has an alcohol group. So all of those having that oxygen means that it can hydrogen bond with water. So it's going to be polar. It's going to want to be able to interact with water if it gets the chance. The last group, and this is the last 5, so there's a total of 11 that are polar. These ones are polar, but not only are they polar, but they're even charged. So if we zoom in and look at them, so you see the aspartate actually has oxygens and it even has a negative charge. Same thing with glutamate, charged. Cysteine and arginine, lysine and arginine, nitrogens with positive charge, plus just having those nitrogens. And these ones are pretty big as far as they've got 4 carbons on them, but they've got that charged nitrogen down on that end, and that's going to make it want to interact with water. So these are also going to be polar. Histidine, double nitrogen on it. So these ones also polar. So there's really 11 out of 20. So about 9 of them are nonpolar, about 11 of them are polar. These make them hydrophilic.
Do we need to know all of them?
No.
Okay.
No, if you really enjoy it, you could take a class in biochemistry. That's what my master's degree is in. And at one time in my life, I could actually draw all of these, knew all their characteristics and everything. But no, that is a class in and of itself. So with these, if you just remember that the R groups, that some R groups are mostly hydrogen and carbon, those are going to be your nonpolar groups. The other ones, if they have oxygens or nitrogens, then they're going to want to interact with water. They're going to have a polar characteristic. And that's going to come back. Because when these form links, just like 2 sugars get linked by removing water, we can do the same thing here in forming a protein. So one side of the amino acid will react with the other side of another amino acid and remove water. So remember, we called that a condensation reaction. Condensation reactions that link 2 molecules together, pull a hydrogen, pull an alcohol group off of the 2 in order to join them. We did that when we were talking about this first example they did. This is what happens with sugars. So one side you have an OH, one side you have an H. It pulls those off and then forms the link between them. The middle one is actually how fats are linked together into triglycerides. Long fatty acid chains get linked together. But this last one forms what they call a peptide bond. So in a peptide bond, the oxygen gets pulled off of the carbon with the two oxygens. So this oxygen is going to get pulled off, and two hydrogens get pulled off of the nitrogen, and that's where your water comes in. And that's going to form a link between one amino acid to the next one. And remember that that's called an amide. That type of functional group, peptide bonds, are what link amino acids together. So we can do this. There shows this one up close, a little bit better, okay, a little larger. So in this amino acid, and remember, see that this amino acid differs from this amino acid. The only part that's different is the part I circled. All the part, the NCC, that's all the same. So all of the bottom part is exactly the same, except for this and this. So that's what makes one amino acid different from another. That's the R group. But the R group is not really involved when the links form. So when the links form, it is the carbon on the end, the carbon with the two oxygens, links to the nitrogen on the next amino acid. So it's these two. We're going to pull the oxygen off of here. We're going to pull two hydrogens off in order to form water.
And the carbon links to the nitrogen?
And then the carbon gets linked to the nitrogen. So this carbon is going to get linked to the nitrogen. So see this bond that's here. We pulled two hydrogens off of the nitrogen, the oxygen off of the carbon, and it forms the peptide bond. So now you would say that alanine is linked to valine. Just two amino acids linked together. But it's pretty rare that you just see two amino acids. Instead, you end up seeing lots of amino acids. So in this, if you follow along, do you see how it's like you got a chain and then you got stuff hanging down? The chain, they call it the backbone. The chain is NCC, NCC. Remember I said that nitrogen, carbon, carbon, that's the same. Nitrogen, carbon, carbon, nitrogen, carbon, carbon. Each NCC is an amino acid. So you could kind of go along and say, okay, well, this is one, this is one, this is one. So NCC. So there's a total of how many amino acids in this chain?
8.
Yeah, there's 8 of them total. Okay, so if you would split, you would just split each C from the next N. So this is an 8 amino acid protein, oftentimes called a peptide, which just means a little protein. And which ones these are depends on the pieces hanging down. So the ones hanging down, these are the R groups. And so you could like look on a chart and you could look and see, okay, which one has a carbon and another carbon and then 2 oxygens attached. So you would be able to find that and identify what that amino acid would be. And then it's linked to this next one, linked to the next one. So this they call the primary structure. It is the order of the amino acids. Each amino acid is linked to the next one by a peptide bond. And we'll actually talk about how peptide bonds, how proteins are put together in chapter 11. But now when you notice this, do you notice these oxygens sticking up? Those oxygens, and remember those oxygens are going to have non-bonding electrons. So the oxygens have non-bonding electrons. But there's also in the backbone, there's also nitrogen. And remember, nitrogen's in group 5. So it makes 3 covalent bonds, but it's got a pair of non-bonding electrons. So these are like this. So you've got those extra electrons. So when you take this protein and you put it in water, it doesn't just stay straight. because it's going to begin to fold to try and interact. First, the interaction occurs with what they call secondary structure. Secondary structure is when those double bond oxygens come close to the nitrogens. The nitrogens in the backbone form hydrogen bonds with the double bonded oxygens. So if we blow this one up so that you can look at it just a little closer. Can you see that? Can you see how this carbon double bonded oxygen hydrogen bonds with the nitrogen? And so you start to get a twist, and the twist lines those up. The dots, those are the hydrogen bonds. So this hydrogen bond, this hydrogen bonding, and what it does, this reminds me of a slinky. Okay, so it starts to form this coil that is sort of like reinforced or stabilized because of the hydrogen bond, but really forms this little helical slinky. So if you grab these ends and you pull on them, are they just going to snap? No, what are they going to do? They're going to stretch. Okay, so this arrangement actually gives you a little bit of stretch to a protein. So you're not talking about like a piece of string that you pull on it and it would snap. This is going to have a little more give. It's going to give a little more elasticity, and you can kind of see that this like coil kind of could flop back and forth, but not actually get damaged. So hydrogen bonding, and just think of it as secondary structure is hydrogen bonding of the nitrogens and carbons in the backbone that form the peptide chain. So it is going to allow this strand to actually stretch and recoil like a slinky. So it has some give to it, doesn't snap as easily. So they might look like the helix, and so they call this an alpha helix. And it's just the twisting of it, so that's where the name comes from. So they call this an alpha helix. The other way that the backbone can be folded is it can be folded back and forth, and reminds me kind of like an accordion. So it gets folded back and forth. So if you follow, this is this is one part of the chain, and here's the other part of the chain. So there kind of does this. It folds back and forth and back and forth, and it's going to give that accordion effect. You know, like you used to take paper and fold it back and forth and make a fan. Okay, so you would hold it on one end and it would like spread out so that then you could fan yourself. Well, that's kind of the same thing. So it has that give to it, still helps to improve sort of the stretchiness or elasticity of it. So you can see either alpha helixes or beta, what they call beta sheets or beta pleated sheets. That's all because of the backbone. The nitrogens and the oxygens in the nitrogen, carbon, carbon, nitrogen, carbon, carbon chain, they'll either fold into a helix or they'll fold back and forth on themselves into this pleated sheet. In fact, some proteins will actually have areas where they have alpha helix, and then areas where it'll actually flip back on itself and look like a pleated sheet. Pleated sheets, do you remember the ribbon candy? Like, I don't know, like my grandmother had them. Like at Christmas time, when you look at it, it was this like folded candy. It was about like this thick and hard, and it had that like folded look to it. That's what I always think of when I think of this. But this is what they call secondary structure. So secondary structure is going to help to reinforce the strand, but also give it some elasticity so it doesn't break super easy. Tertiary. Okay, first is just the order of the amino acids. Second is the helix or the folding that you see. Tertiary structure comes into play with the differences in the R groups. I said some R groups are polar. Polar R groups, if you had a protein and you put it in water, where will polar R groups want to be? Well, they want to be inside or on the surface if they're in water. Polar R groups, they're hydrophilic. They will want to orient themselves onto the protein surface because it's in a watery environment. So polar mixes with water. So polar R groups want to be on the outside. So they want to be near water. They're hydrophilic. Non-polar though, remember I said there's 9 non-polar. The non-polar R groups, they're not going to want to mix with water because they're like oil. And so they will actually bury themselves into the inside of the protein. The hydrophobic, they want to be on the interior of the protein. So the more hydrophobic areas you have, the more sort of twisting the coils and the pleated sheets end up happening, because they don't want to interact with water. So they're going to like bury themselves and create folds and little pockets within the protein. What about if you have those positive and negative charge? Right, so I showed you like a nitrogen can have a positive charge. Some of them had a carbon with an oxygen that had a negative charge. Plus and negative R groups make what they call salt bridges. They're going to be really attracted like magnets. So they like have like ionic interactions. The positive R groups and the negative R groups are going to want to be close together to each other. And this is what you end up with. So this is what tertiary structure does. Can you see helix in this? Can you see some coils? Can you see areas where it's folded back on itself? Okay, that's pleated sheet. So this is alpha helix. This is some pleated sheet area. And that's secondary. So alpha helix and pleated sheets are secondary levels. But then see, it doesn't just do that. Now it's like flipped over and twisted up and around. So you get this movements that occur because here's hydrophobic R groups. They all want to hang out together. So they may actually cause 2 parts of a protein to come together because they want to be together. Same thing, here's some salt bridges over on this side, positive and negative charged R groups. They're going to want to stick together. And so that caused this, instead of it just being a helix, it was a helix, then gets a bend in it, and then another helix. So it starts to cause this 3-dimensional shape. This is tertiary structure. Here's hydrogen bonding between between the backbone and the pleated sheet. Here's polar R groups wanting to interact with water. So the polar ones want to be on the surface, non-polar ones down deep. This is 2 polar molecules. 2 polar R groups want to hang out together because they're similar. So tertiary is going to give you more 3-dimensional shape. The more variation in the R group type, the more diversity in the molecule. So there are some proteins that have alpha helix shape. So they're pretty much like this. And it's because their amino acids are hydrophilic, so they'll interact with water, but they'll like wrap themselves so that the R groups stick outward, and then you get the hydrogen bonding of the backbone. So you'll just get a long coil. It's when you start throwing in nonpolar molecules or charged R groups that you're going to start to see these shifts or these movements. When it's like this kind of shape, they typically call this a globular protein. Can you kind of appreciate why they would call it a globular protein? Because it kind of looks like a glob. It looks like somebody just took like random yarn that had all different kinds of twists in it, just kind of pushed it all together. They call this fibrous, Can you see that that looks longer like a string, more like a strand? So fibrous has less tertiary structure. Globular has more. So looking at this, and I really should change the color from blue, but looking at this, you tell me, what is this R group? Would this R group be polar or non-polar? Polar. If you see an oxygen or a nitrogen in the R group, best odds are it's polar. So what about the next one?
Charge.
Charge, so it's polar. Nitrogen or oxygens, it's polar. What about the next one? That's the non-polar. See, nothing but carbons and hydrogen. So this one's non-polar. That one, polar. That one, non-polar. Polar. What about that one? Do you remember? This is just carbon, carbon, carbon. It's going to be non-polar. No oxygen or nitrogen, non-polar. Okay, what about the last one?
Non-polar.
Non-polar. All right, so this is our 8, right? This is the same one as what we looked at before. So this is 8. So which ones are you going to find towards the surface? All the polar ones. Okay, so the polar ones, I'm still going to get potential of alpha helix or beta pleated sheets, but the R groups is going to want the polar molecules are going to either interact with each other or interact with water. So the polar R groups are going to fold themselves so they're like on the outside surface. Or they're going to be together. So they will group together because they're like each other. Like mixes with like. But those non-polar ones, and I have 1, 2, 3, 4 of them. Out of this 8, 4 of them are non-polar. And those are the ones that are really going to cause a change in the shape. So the non-polar are going to do what?
Stay in the protein.
They're going to be down in the inside. They're going to form almost like a core. So in this one, this one, this one, and this one. Those four non-polar ones are all going to end up like that. So instead of seeing it nice and flat, you're going to end up, those red regions, the non-polar regions, they're going to be in the center. And then the polar areas can be looped towards the outside, coiling towards the outside, creating pleated sheets, but they're going to have easier time interacting with water because of that hydrophilic nature. So that's tertiary. A couple of examples of tertiary. Sheep. I like their little face. Sheep. Keratin and collagen. So keratin and collagen, they actually have more than one protein strand, which we'll talk about in a minute. But when you look at that strand, what do you see? That strand looks like a what? It just looks like an alpha helix. Doesn't it just look like a coil? This is collagen, is this one, and that picture is like a colorized MRI that is looking at a knee. So this area, all that yellow, is looking at cartilage. Collagen is the most abundant protein in your body. Collagen is what makes up all of the anchoring tissue. So underneath your epidermis, the dermis of your skin, the reason that it stays on is because the dermis is almost all connective tissue. Dense, regular, dense, irregular. Then you've got the cartilages. That's all collagen. Collagen with some water to make it kind of spongy. When you're talking about bone, bone is all collagen, but it's had calcium added, which makes it rock hard. So all of those connective tissues except for blood, all the connective tissues all fall into this category. They all have this type of arrangement. So collagen looks kind of like multiple chains of alpha helixes twisted together. If you look at a piece of thread up really close, that's what it looks like. So thread is these strands of cotton that have just been wrapped and then wrapped around each other. And that adds the strength. So tendons and ligaments, like what actually helps to allow pull on bones, those are tendons. So you have muscles that contract, and then you have the tendons that pull on bones. When you're talking about the ligaments are what link bones to bones together. That's all collagen. So they are fibrous, long, thread-like. They tend to be insoluble. They don't dissolve in water. They tend to stay. I like to think of them as the big anchors. These are your ropes, and they look like thread. They look like string. Collagen, the most abundant protein in the body. Is a fibrous protein, very resistant to damage, very durable. Keratin, the one that they showed with the sheep, keratin is what your external surface of the skin, the epidermis, your nails, your hair, that's what keratin is. And if you look at the two, do you notice how keratin looks like it's packed tighter? It's kind of like smaller looking compared to collagen. It is. It's actually wrapped so tight, it squeezes water out. So keratin is a waterproofing protein. So they show sheep because sheep can stand out in the rain, and the keratin that makes up the lanolin in their coat, just like water just runs off of them. So it like literally won't soak in, it just runs off the surface. So it's a waterproofing protein. It's a little smaller than collagen, but it resists any water absorption. And that's how it keeps it dry. So that's why your nails, like they're dried out, why the skin doesn't wash off when you take a bath. So it's all that waterproofing that pushes or keeps water from being able to come through, but those are fibrous. Globular, though. Globular proteins, if they have this arrangement, globular proteins, their shape is important. So they're not as sturdy. Their shape determines what their function is. Enzymes are typically globular. So remember with enzymes, I like kind of drew like the little E laying down, but I did it like the little sort of like helix looking appearance. Antibodies that have the responsibility of binding to things that are foreign, it's their shape that's important. Receptors. So remember like the receptor for insulin? So that's on the surface of your cells and insulin fits. It's the shape that's important. So with the globular protein, they're actually more sensitive to damage. Because you could take this globular protein and like shake it up. If you disrupt it, then all of those interactions can be lost. Globular proteins, it's their shape that allows them to function. They're more sensitive to damage compared to fibrous. So the last one, it's Is it my phone or my watch? Which one's going on? The last one. The last one's quaternary, which just means the fourth level. So primary is the order. Secondary is the helix or the pleated sheets. Tertiary is this folding that you get because of the R groups. Quaternary occurs if you have more than one protein. So if you have greater than one protein, and you'll end up forming what they call, what I think of really as a complex. It's going to make a protein that's more than one strand, but altogether those strands have a special function. Not all proteins have that, but now keratin and collagen do, right? You can see like with collagen, there's the gray, the orange, and the purple looking strands. So there's 3 strands. They don't have a whole lot of folded shape, but it still is a complex. It is still more than one protein strand in order to make it. Keratin, all the strands are the same, but it's still more than one strand. So those would have quaternary structure. The best example of the quaternary structure has always been hemoglobin. So that's what this is. And this kind of like summarizes all 4 levels. So this is just the order of the amino acids. So amino acid 1 to amino acid 2 to amino acid 3. So whatever the chain, the primary structure is just the order of the amino acids. B, the secondary structure. So we call this what? What is that secondary structure called? Goes around the helix. So it's the alpha helix or the pleated sheet. Remember, that's going to give you a little bit of stretchiness, elasticity. Tertiary structure. This is the R groups. So that's what's causing more folding. It's what causes stuff down in here is going to be what? More likely than not, the R groups down in there are going to be the nonpolar ones. I'll do them in yellow. So the ones that you find buried down inside, those are going to be the nonpolar ones. The ones that you find all the way out on the outside are going to be the polar charged ones. Hemoglobin is actually 4 proteins. Can you see that there's 4? There's 2 that are colored gray, 2 that are colored purple. Hemoglobin's kind of special because it not only is 4 proteins, but it's got 4 of those other things. Does anybody know what that is? See this? What does hemoglobin have? It's in red blood cells. It's the reason why red blood cells are red. What do you have to have in your diet to make red blood cells? Iron. And that is what those are. There are 4 iron atoms in one hemoglobin protein, and it is the iron atom that binds oxygen to be able to transport it. So remember we said that oxygen diffuses into the blood at the lungs. Hemoglobin, the iron actually attracts it and holds it, carries it along. When your blood's oxygenated, what color is it?
Red.
Bright red. Oxygenated blood, or is arterial blood, bright red. So after blood goes to, and it's because the iron gets oxidized. So the iron grabs oxygen, which makes it oxidize, which is the reason why it's a bright red color. But what about the blood that's in your veins? So when you look at them, see how dark they are? They're dark because the blood that's in there is deoxygenated. So when you take the oxygen out, then that actually causes them to get reduced. And that's going to cause them to get really dark in color. If you've ever seen like venous blood is like almost like red black. It's a really dark color. As soon as it hits the air, it gets lighter, but it's a very dark color.
Are those like the vertical veins and stuff?
Well, any vein other than pulmonary, any vein is going to be low in oxygen because arteries take blood to your tissues, veins bring blood back. And after it's gone to the tissues, it's been deoxygenated. So the reason in the color is all has to do with the content of oxygen on each of those irons. So you need to have iron in your diet or else you end up with what? So low iron, anemia. And how do you feel?
Sleepy.
Tired, low energy. You need oxygen to make energy. That's the whole reason for carrying oxygen is your body cells use oxygen to make ATP. So if you have low levels of oxygen, your cells are making low levels of ATP. If you get up really fast, what happens? Yeah, you get like flashes of light, whoo, whoo, going down, that kind of sensation. That can all be because of low iron. Okay, so there's the 4 levels of structure. Primary is just the order. Secondary, the helix or the pleated sheets, giving you some stretchiness. Tertiary, folding. Hydrophilic likes to be on the outside. Hydrophobic is going to want to be on the inside. And if it's got charged groups, positive and negative, positive nitrogens, negative oxygens, they can make salt bridges. And that's going to give you this, that 3-dimensional structure. Lots of variation, lots of 3-dimensional structure, and you can end up with a globular protein, which looks like a big glob. Okay, and it's just because lots of hydrophilic, lots of hydrophobic, lots of folding to it. So I told you that they're kind of sensitive. Globular proteins, you can change their shape by affecting their environment. So one of the things that we did in lab is we used an acid and a base. We did this when we did the lactase enzyme treatment. So you took a beaker with lactose and you added acid to it or you added base to it. And then we measured to see how active the enzyme was. For the people that did the acid treatment, none. Like nobody got in, everybody had zeros over the 45-minute span. So that tells you that that enzyme does not do well in an acidic environment. So if you add acids or bases, sometimes even if you change the salt concentration, that will cause something that's folded like this to unfold. That's called denaturation. So denaturation is when you disrupt the tertiary, quaternary structure, in some cases, even the secondary structure. Most of the time it's tertiary and quaternary. So you take something, especially globular proteins like enzymes, change their environment and you can change their shape. So the first one, heating. Can you give me an example of a protein that starts off as a colloid and it's all gooey, and then when you're done cooking it, it's a solid? Egg white. Egg white is almost entirely protein. So this is, does it go back? No. You can't take that egg white and turn it back into the gooey stuff. That's a permanent denaturation. Another example of this, whipping cream. If you've ever whipped cream, same thing. You take the mixer and you put it on high. And so it is like spinning and agitating so much that it will actually cause the proteins in the cream to denature. And so it goes from being a liquid to being a kind of solid. Like you just keep doing it until you get the little peaks and then you know that it's done. If you make meringue, like if you like lemon meringue or key lime meringue, meringue is doing the same thing to egg whites. You just beat them until they actually form, they become like semi-solid just from mixing them. So one of my friends used to make biscuits, but she always put some vinegar in her milk. What happened to it? Anybody ever do that when they cook? Like literally a half a teaspoon is all you need. What does it do? It causes it to curdle, like it makes the milk look lumpy, okay? It's called clabbering, which is like a word I'd never heard of until I moved to the South. I was like, clabbering, wow. So look that one up. But what she did is she added acid and it caused the milk to denature the proteins, and that actually makes the biscuits better. It makes them softer, it helps them to rise the extra acid in them. So she made like the best biscuits. My biscuits look like hockey pucks and taste worse. Detergents. One of the reasons that you use soap, okay? Soap, remember, has polar and non-polar parts. So soap is going to interact with a globular protein, and it would cause non-polar R groups to go into the soap. Polar parts would be sticking out of the soap. So it's going to cause it to unfold. It's going to change its shape. So detergents help in denaturing proteins. Heavy metals. This is silver, is Ag. PB is, anyone? PB. From the Latin plubnum, which doesn't help you. Anybody know what PB stands for? No. Do not eat this. This is lead. Okay? PB is lead. So Ag is silver, PB is lead. Hg? Mercury. Okay, you should know that one. So that's, but PB, just to tell you, so lead, up until about 25 years ago, lead was commonly put in paint because it made it stick to the wall better. So if you painted with lead-based paint, it would not peel. Like it would actually stay. So you wouldn't have to paint your house every 5 years. You could paint your house every 25 years. That's how good it was. So yeah, people would like slap lead paint all over the house. Problem comes into play is that lead actually, as a salt, tastes sweet. It's not usually that they lick them, but if there's any peeling paint and you got a little kid, what are they going to do?
Eat it.
They're going to peel it, right? And so if they put their hands in the mouth, which is like all what little kids do, like they're super oral. So everything they pick up and everything goes in their mouth. They put it in their mouth and it's sweet. So it has like a sweet taste to it. And so then what are they going to do? Yeah, they're going to sit there and peel the paint and eat it. But no one's telling them not to. And it's really like, oh, this tastes pretty good. Problem comes into play, silver, mercury, and lead. That these, lead in particular, actually concentrates in the brain, and it will actually cause denaturation of proteins associated with neurological development. So what can you end up with? Mental retardation. So that is what they found as they started seeing these learning disabilities in kids and then started looking at the environment. Well, where is this? Like, what are we looking at? And that's what it was. So that's part of the reason why they took lead-based paint, because you get to think even outside, like when I was a kid, we used to go outside and dig in the dirt, like around the house. So if you are painting with lead-based paint and you would go and like scrape it and then repaint, all the scrapings, did you clean all that up? If it's outside, would you bother cleaning up scrapings? No. So you would actually have like higher levels of lead in the ground around the outside of the house. And so anybody like playing in the dirt and stuff like that, all going to come in contact with it. So that's the problem with lead. Tastes sweet. So peeling paint. Kids eat it. But it ends up having a negative effect on brain development. So it actually causes proteins that should be forming are blocked from forming. So you'll start to see that there'll be learning disabilities that begin to show up. Bad thing is when it gets to a certain level, trying to actually get it out is really difficult. So if you get to the point those learning disabilities may not be overcome just by removing them from the environment. So that's why most people now, so if you buy a house, you have to state when it was built, and you also have to like know whether or not there's any lead-based paint. And if there's lead-based paint, they don't actually want you to sand and remove it. They want you to paint over it to seal it, to make sure, especially if it's inside the house, is to make sure that it gets sealed in instead of removed, just because of that just makes even more dust.
The same thing with asbestos floor.
Yeah, with asbestos tiles and things like that. If you can't remove it without making dust, then you'll just seal it in, because the dust in and of itself is toxic. Okay, so all just examples. So heating, shaking or stirring, adding acids or bases, using soap, all help in denaturing proteins. Maybe you want to denature bacterial proteins, and so that's where the soap actually helps with that. But if you're not wanting to denature, then sometimes you have to be a little careful what you do with your proteins to keep them from happening. This is kind of like now about function. this sort of leads us to this, like the last big topic. Like all of this is nothing but theory stuff. But there's 8 big protein functions. 8 functions. And I tried to group them together. Like this page has 3. So 1, proteins might be messengers. Messengers are carrying information. And the best example of a messenger is a hormone. And in our picture, where's the hormone? Where's the messenger? Insulin. So here's the messenger. Because insulin tells your body cells what? That there's sugar outside of the cell. So the sugar's sitting outside of the cell. Notice the insulin comes along and it binds here. So the insulin binds. Where it binds is number 2. That is the receptor. So the receptor, the shape of the receptor, can interact with a messenger. To send a signal to the cell. So you have the messenger, insulin, binds to the receptor on the surface of the cell, and that tells this to happen. So what's happening with the blue? What's happening on the right? So glucose is going from outside in. You have to have the messenger, or else the receptor never does anything. But once the hormone binds the receptor, then that signals to the transporter, and that's our 3rd one. So this transporter is an integral protein. Remember, integral means it goes all the way through the cell. So an integral protein acts as a channel. It's a transporter to bring that polar glucose into the cell. From high concentration to low concentration. So three functions of proteins right here. Okay, you have the hormone, which is the messenger. You have the receptor, which binds to the hormone, and then you have the transporter that allows molecules to go in or out. 4, carriers. So we talked about hemoglobin. So hemoglobin is the carrier of oxygen. What was another carrier we talked about? Something that was responsible for transporting in the previous lecture. What's the thing that transports cholesterol and lipids? 2 things, the good and the bad. LDL and HDL. They're low density lipoproteins and high density lipoproteins. So these 2 carriers, they're part protein. They interact with cholesterol and other fatty molecules to transport. Just like hemoglobin transports oxygen, so you do have a number of like transporters in the blood that are responsible for moving things from one place to another. So that's 4. 5, protection. This is like whenever you look at an antibody, it's always drawn like this. I'll just tell you now. Like in an AMP, if you ever talked about antibodies, they look like Ys. You're like, so there's a whole bunch of Ys that are made by B cells. Okay, so antibodies are proteins, and their shape here allows them to bind to foreign substances. So if you have a foreign cell, this antibody binds to it. So there is actually the shape of the antibody allows it to stick. And the great thing is if you have a little foreign cell and antibodies are attracted to it, recognize it, and bind to it, But this cell, this little foreign cell, is trying to be like super stealthy. Is that going to be able to do that? If suddenly it has all of these antibodies stuck to its surface, is it going to move very fast? No. It slows it down. Is it going to be able to grow really fast? No, because it's kind of like now got like all this like antibody sludge all over it. The antibodies also act like a flag. They also attract macrophages. Antibodies are like, hey, this is foreign, come and eat it. So that's like another big benefit. It slows down foreign cells and it brings your white blood cells into the area. So it acts as like an attractant for the rest of your immune systems. And it has to stick. Don't want it sticking to things that are not foreign, which happens in some autoimmune diseases. Slows foreign cells and also attracts macrophages. So it really just helps to speed up how fast your immune system can respond to something being present that doesn't belong there. And it's this shape right up here. This binding area is unique and different. It's specific for that foreign cell. It won't bind to human cell or body cells, but it will bind to things that are foreign. But they always draw it looking like a Y. 6 and 7. 6, contraction. Okay, so you find contractile proteins where? In your muscles. Skeletal, cardiac, and smooth. Smooth doesn't have them arranged as like tightly packed so that you don't really see the striations. Actin and myosin. So when this gets stimulated, the actin proteins are pulled on by the little myosin proteins. Can you see how they overlap? So this is in contracting, going from here to here. So before, when they're relaxed, the muscles are like this. But when they're stimulated, the overlap increases. And so that happens with each little stack of actin and myosin. And that allows for contraction. And then when the contraction is done, it slides back. So it goes back to that resting state. So this allows for movement, for pumping of the heart. 7, collagen. It's the anchor. When I think about fibrous proteins, I always think about collagen because it looks like a string, because it looks like thread. Good example of this, your tendons and ligaments. Tendons anchor muscles to bone, ligaments anchor bone to bone. So you can think of it like it ties this to this. And it forms embryonically. Like if you tear a ligament, they have to go in with screws and like cadaver tendons or cadaver ligaments to reattach them. Like you can't go and stitch this back together. Like so they are formed embryonically and then they get stronger and stronger as you grow. Another one that's kind of interesting is one called elastin. So elastin has a lot more beta-pleated sheet to it compared to collagen. And so can you see that you can take elastin and you can do this? So it's like a rubber band. So you can stretch it and then it bounces back. And so there's a lot of elastin in the skin and that's why you can like pull it and it goes back. Okay. If you look at like little chubby baby cheeks and you like pinch them, okay, they go there like really plump and soft and they go right back. But what happens is you start getting older. Does that keep happening? No. And that's what happens with wrinkling. So elastin should look like this, But over time, the elastin's not quite as ruffled as maybe it used to be. And so that's when things start hanging. And you're like, oh no. That's wrinkling. Normal occurrence. Speeds up though with the exposure to ultraviolet light. So I don't know if you've ever had any friends that that tanned all the time, that were super, super brown. And then when they get into their 50s, what do they look like?
Leather.
They look like leather. Well then, that's because of damage to elastin underneath the skin. Causes like premature aging and wrinkling to speed up. Okay, so movement contraction is number 6. Anchoring, number 7. Last one, number 8. These are the 8 big functions of proteins. Number 8 are the catalysts. And we have talked about enzymes. So enzymes, remember, reactions would occur. They would just take place so slow, we would never survive. Tertiary structure, really important, that folded appearance. There's their picture. So in this reaction, that little cleft, this space right here is just the right shape for glucose to fit. So this enzyme allows glucose to like fit. I think of it as looking like it's in a little pocket. So it fits in that pocket to speed up this reaction. So it takes glucose and it adds a phosphate to it. So that reaction brings those two together. So remember, in doing this, the way that they speed things up is they take two reactants or two substrates and put them together. So if we have A and B, we just have to wait for them to float together and hit each other. But if an enzyme takes A and takes B and puts them together, close together, and in the right orientation. Remember, I think of Legos. Like Legos only stick together if you put one on top of the other. If you put them side to side, they're not going to stick together. So the orientation is important. Same thing here. I kind of like that picture. This is your enzyme. Notice that you could put the orange and the green together all you want, but unless you put them together like this, the reaction won't happen. So that's what the enzyme does. So it takes two substances or takes reactants, brings them together in the right orientation for the reaction to occur. This shows sometimes an enzyme speeds up the breaking of a bond. And so in this one, do you see how, well, this one's actually forming, where we're breaking, we're taking this phosphate and we're transferring it over. So you see that one of the phosphates ends up on the glucose, the orange hexagon, tracked over. So the enzyme puts them together so those bonds are super close. Okay, so that it's not difficult to just move it from here to here. If they were over here, the odds of them transferring really slim. So the right proximity, the right orientation, and that helps to decrease the bond energy. And remember, that's the activation energy. So if we decrease the activation energy, that is going to speed up the reaction. So we'll talk about concentration, pH, and temperature to finish this up. We've got to talk about inhibitors as well, but I've only got like maybe another 15 minutes in this. So it's really not a difficult chapter. Just go through like the structure part of proteins, about the 8 functions in proteins and organizing them that way. Look back at denaturation, examples of globular and fibrous proteins, all of those like major things that we've talked about tonight. We'll talk about ways that you can change or affect enzyme activity, and then we'll go on to chapter 11 next Monday night. So I hope you have a really good Thanksgiving. No class on Wednesday. Yes, no class on Wednesday. No. You can go home and do all your pre-Thanksgiving baking. Yay.